Dear students, welcome to the Zoological Society of Assam. In today's lecture, let us make an attempt to know about another important topic on arthropods, that is respiration. I would like to offer my sincere thanks to all the viewers of my first presentation in this lecture series. Before going to the detail, I would like to remark here that this presentation is a part of the initiative taken by Zoological Society of Assam to communicate open educational resources to the undergraduate and postgraduate students of biological science in general and zoology in particular. As we know, the primary aim of respiratory systems in arthropods and in other animals is to deliver oxygen from the air to the tissues and to transport carbon dioxide from the tissues to the air. The phylum Arthropoda is the largest group of animal world. They live in different types of habitats, including terrestrial and aquatic. Accordingly, the arthropods have developed different structures and mechanism of respiration. With this presentation, we will understand the primary respiratory organs, their structure and mechanism, such as book gills, the respiratory organs in aquatic arthropods, book lungs, respiratory organs in terrestrial arthropods, tracheal system, the primary respiratory organs of insects, gaseous exchange in aquatic insects obtaining oxygen from water, and aquatic insects obtaining oxygen from air. And finally, gas exchange in insect eggs and gas exchange in endoparasitic insects. Let us start with book gills, the organs of respiration in aquatic arthropods. Book gills are characteristic respiratory organs of aquatic arthropods, especially crustaceans, though this can also be observed in some arachnids such as limulus, or commonly called as the king crab, as shown in the figure. These are present in different places of the body, based on which they can be of various kinds, such as podoprangs, also called the foot gills, as they are attached to the coxa of an appendage, arthroprangs. These gills are attached to the arthrodial membrane which joins the leg to the body of a crustacean. These are also called joint gills. And the pleurobranchs, the side gills attached to the pleural membrane of the body segment bearing the limb, are termed as the pleurobranchs. Structure of book gills. The book gills are often flap-like or crescentic in shape. Why they are called book gills? They are called so because they resemble a book. Each gill is made up of several thin structures called gill lamellae or gill plates. These are the gill plates or gill lamellae in a limulus. These are arranged like the leaves oblique pages of a book at the right angle to the gill base, which is also called the gill axis. Mechanism of respiration through book gills. Book gills, like fish gills, are bathed in water and supplied with blood vessels. The exchange of respiratory gases takes place between water and blood flowing through the gill lamellae. The deoxygenated blood is carried to the gill lamellae with the help of afferent branchial vessel. During the flow of blood, gaseous exchange takes place with water, which enters the gill chamber in the form of a current. The thin membranes of the gill lamellae act as an excellent surface for diffusion of respiratory gases. Consequently, oxygen from the flowing water diffuses into the blood, while carbon dioxide from the deoxygenated blood diffuses in the water. Similar to those of book gills in aquatic arthropods, book lungs are the organs of respiration in terrestrial arthropods. These are always in pairs and considered to be evolved from booggills as an adaptation of terrestrial mode of life. As shown in the diagram, booglungs are often located in the abdominal segments of the arachnids. Booglungs are blind in pocketings with highly folded inner linings across which gases diffuse between hemolymph and the air. The structure of booglungs. As shown in the figure, a booglung is consisting of two parts, a dorsal chamber and a ventral chamber. Dorsal chamber is also called the pulmonary chamber, formed by the invagination of the ventral abdominal wall. It consists of approximately 150 leaf-like lamellae, each of which is lined with the cuticle. Ventral chamber, also called the atrial chamber or atrium, is the non-folded part of the booglung and is filled with the air. Mechanism of respiration through booglungs. The movement of air in the booglungs is controlled by certain muscles attached to them. These include atrial muscles and dorsoventral muscles. The contraction of the muscles results in the compression of the booglungs. As a result, the air is forced out from the interlamellar spaces to the atrial chamber, from where it is released outside through stigma or spiracles. On the contrary, the booglungs regain their shape by the relaxation of the muscles, and the air enters the atrial chamber through stigma reaching interlamellar spaces. In the figure, this is the interlamellar space, and these are the lamellae of lungs, and this is the atrium or atrial chamber, and this is the spiracular opening or stigma. The position of booglungs and booggills. As we mentioned earlier, the booglungs and booggills perform similar function, but their position is different. The booglungs of arachnids are made up of alternating air pockets and hemocoel tissue, shaped like a stack of books and are internal in position. On the other hand, the booggills of crustaceans are also similar to booglungs but are external, so that gas exchange can occur with the surrounding water. Now we will try to understand the primary respiratory organs of insects, the tracheal system. Gaseous exchange in a majority of insects occur through a system of the air-filled internal tubes, the tracheal system. Tracheal system simply are the air-filled internal tubes. An air-filled cuticle-lined invagination, the finer branches of which extend to all parts of the body. Oxygen is carried by the trachea in the gas phase directly to its site of utilization. While most of insects appear to have intracellular hemoglobin and some groups of insects have hemocyanin in the hemolymph, the dominant pathway of gas exchange is by air-filled trachea. The tracheal systems provide a lightweight, high-capacity oxygen delivery system that enables the highest rate of gas exchange in animals and recovery from anoxia by diffusion. The trachea opens to the exterior. With the help of spiracles. Usually two pairs of spiracles on the thorax and 7 or 8 pairs of spiracles on the abdominal region in insects. This is the diagram of an insect belonging to order Hymenoptera, showing the tracheal system with the position of spiracles. Let us try to understand the movement of air in the tracheal system, or simply gaseous exchange through the tracheal system. The gaseous exchange in the tracheal system is primarily by diffusion and ventilation. It takes place in the following steps: alternate contractions and expansion of the insect abdomen lead to the changes in the diameter of trachea, which results in the movement of air in or out of the trachea. During inspiration, air enters the trachea through the spiracles and passes through the tracheoles to the fluid-filled tips, where it gets dissolved in the fluid. Now the oxygen diffuses from the tracheoles into the body cells, whereas carbon dioxide diffuses from the cells into the tracheoles. During expiration, carbon dioxide-rich air is released outside the body through trachea. However, carbon dioxide can also diffuse outside through the cuticular covering of the body. Now come to the aquatic insects. Gaseous exchange in aquatic insects. Aquatic insects obtain oxygen directly from the air or from that dissolved in the water. The insects which obtain oxygen directly from the air necessitate some semi-permanent connection with the surface or frequent visits to the surface. Insects that obtain oxygen from the water nearly always retain a modified tracheal system, so that the oxygen must come out of solution into the gaseous phase. Now I want to explain briefly about the structures involved in case of aquatic insects for obtaining oxygen. Firstly, the aquatic insects obtaining oxygen from the air. Gas exchange by respiratory siphon. Respiratory siphon, as shown in the figure, is a tube found in larvae of certain insects used for breeding. For example, the larva of hoverfly eristalis belonging to order Diptera. Gas exchange by the air bubbles. Some aquatic insects, such as mosquito larvae, can remain submerged only as long as the supply of oxygen in the trachea lasts. But others have an extra tracheal air store, carrying a bubble of air down into the water when they dive. The spiracles open into this bubble so that it provides a store of air additional to that contained in the tracheal system, enabling the insects to remain submerged for longer period. So best example of the insects exhibiting the air bubbles is the whirling beetles, as shown in the figure. Gaseous exchange in aquatic insects obtaining oxygen from water involves a variety of structures such as tracheal gills, plastron, spiracular gills, blood gills, and respiratory trumpet. Before going to detail of these structures, one important point to be noted here that in all insects living in water, some inward diffusion of oxygen from the water takes place through the cuticle. Cutaneous diffusion depends on the permeability of the cuticle and the lower oxygen tension in the tissues as compared with the water. The majority of insects that obtain oxygen from water do have a tracheal system, although the spiracles are non-functional. This is called a closed tracheal system. Let us try to understand about the respiratory structures found in aquatic insects obtaining oxygen from water. Firstly, the tracheal gills. Tracheal gills are thin plates or filaments-like structures with abundant tracheal supply. The tracheal gills commonly occur on the abdomen but can also be observed on the thorax. The tracheal gills which occur on the abdomen is termed as the abdominal gills, as shown in the figure in an Ephemeroptera. The abdominal gills of Ephemeropterans are plate-like outgrowths. These are the abdominal gills, and these gills undulates continuously to circulate oxygenated water over their surfaces as the insect swims. Tracheal gills are present within the modified hindgut of dragonfly larvae, creating a branchial chamber. Next is the plastron respiration. Certain insect species have specialized structures holding a permanent thin film of air on the outside of the body in such a way that an extensive air-water interface is always present for gaseous exchange. This film of gas is called a plastron, as shown in the figure. Plastron of Aphilocherias belonging to order Hemiptera and macroplastron of Hydrophilus belonging to order Coleoptera. Hydrophilus is a very common aquatic insect found in the freshwater bodies of Assam. Another important structure associated with gaseous exchange in aquatic insects is the spiracular gill. Spiracular gills are found in insects that inhabit running water that is highly oxygenated but also subject to periodical drying. They are present in the pupal stages of several dipterans and coleopterans that inhabit intertidal environment. Here, the spiracular gills in the pupa of a black fly Simulium is shown. Next is the blood gills. Many trichopteran larvae possess blood gills at the hind end of the abdomen. They are also found in similar position in some dipteran larvae. Blood gills have a spacious lumen but are devoid of trachea, or trachea are poorly developed. Their lumen is filled with blood, and thus these are called blood gills. Blood gills are found mostly in the chironomas. The ventral or anal gill in chironomas is termed as the blood gills. Next is the respiratory trumpet. Respiratory trumpets are structures associated with the prothorax of pupal stage in certain aquatic diptera, such as Culex, as shown in the figure. Structurally and functionally, they are similar to the respiratory siphons. Gas exchange in insect eggs. Physiological studies suggest that the egg chorion is a high resistance structure designed to minimize water loss while allowing adequate oxygen delivery. Gas exchange in insect eggs occurs by diffusion through wax or protein layers. The eggs of many terrestrial insects are subject to occasional flooding, and those of many species have a chorionic plastron. elongate projections of the egg chorion that form a plastron on the egg, facilitating gas exchange but minimizing water loss, is called a respiratory horn. Respiratory horns are found on the eggs of several taxa, including Diptera, Hymenoptera, and some Hemiptera. Gas exchange in endoparasitic insects. Endoparasitic insects may obtain oxygen directly from the air outside the host or by diffusion through the cuticle from the surrounding host tissues. In many ichneumoniid and braconid larvae belonging to order Hymenoptera, the tracheal system of the first instar is liquid-filled, and even when it becomes gas-filled, the spiracles remain closed until the last instar. Thus, these insects and the young larvae of most parasitic Diptera depend entirely on cutaneous diffusion. These are the references or suggested readings you can go through. Rupert and Barnes Invertebrate Zoology, 8th edition. Chapman, The Insect's Structure and Function, 5th edition. Bluska, Murray and Shuster, Invertebrates, 3rd edition. Raj and Hazarika, Non-Chordates, 1st edition. This is our book published in this year. And some web materials. Thank you very much for your time in this presentation. Your feedback is highly appreciated. You can contact me at hazarika.rabindra@gmail.com.